are going to get started with the next part of our program here, which is a panel discussion. So I'm going to MC. I'm Amy Elliott. I head up product at FormBio. I'm a molecular biophysicist by training. The questions that I have prepared for this section are really focusing on areas where we have challenges as an industry, places where standardization is going to be really important, and to give you a really nice overview perspective from our panelists on what their challenges are and where we're missing opportunities in technology that has yet to be developed to pave the way for the future. So with that, I'm going to pass the mic down the panel. If you will introduce yourselves, I'd like you to where you are, what your company does a little bit, and then where you live in the sort of life cycle of a CGT drug, please. You have 1 minute each. Go.
I'm Phil Tai. I'm an assistant professor at UMass Chan Medical School. My primary area of research is, characterizing and developing, novel AAV-based gene therapy platforms for human disease. I'm focused on basic biology, so.
Hi, I am Nadia Selami. I work at PacBio. I'm the lead for cell and gene therapy and biopharma applications. And as you've heard already today, we provide DNA sequencing technology, and we're really interested in helping you develop your products and assess the quality of your products.
Hi, I'm Adam Cockrell. I work at Solid Biosciences. I'm the senior director of vector biology there. And we work on a lot of vector design and early R&D, basically, or early research, really, in vector design for various therapeutic programs.
Hello, everyone. I'm Alpha Diallo. I'm the head of the Solution Science team at Form. So in my role, I lead the team driving technical and scientific pre-sale engagements. And we are really focusing on collaborating with our customers, helping them solve the different challenges that they have. We have a lot of capabilities at FormBio using AI, a secure platform, some bioinformatics tools, and we provide all those expertise to our customers. Thank you.
Hi, everyone. Nithya Jaisraj. I'm part of our commercial R&D team at Resilience, which you saw as a CDMO. We do a lot of services for development all the way to commercial manufacturing, and we have a strong regulatory and quality team to really think through as we're developing new assays and new metrics and new needs for AAV. We have that strong team to really walk through that. As part of my role at Resilience, I'm really helping to manage alliances across our different partnerships. I look through to different technologies to really improve our platforms. So instead of us being the builder, looking to partner with folks in the field and how we can actually integrate your technologies into our platforms to make better processes and better outcomes, so we can get therapies to the clinics, to the patients faster.
Thank you. Thank you. Okay, great. So let's kick this off with the first question. By the way, if we get through the session in time, we'll have some time at the end for audience Q&A with this team, and I'm hopeful that we'll have some good discussion there. So the first thing that I want to zoom in on really quickly is the main challenges that each of you are seeing with developing or designing AAVs. So this might be some of the things we've heard about, right, are biological variability, might be how to develop the process that's going to be purifying, enriching, whatever downstream. It might be, designing short hairpins that are actually going to package. So what are the things that are critically challenging for you where you live?
So because I just focus on basic research, right, we also partner with a variety of industry as well as academic researchers. So I have seen a variety of different vector designs. And since these type of technologies have come out, we've seen, range of heterogeneity, right? And so this is what everyone's sort of very much interested in, right? And so I think at least what FormBio is doing right now is, trying to assess what causes these truncation events. We used to think it was just secondary structure and GC content, and based off of Adam's talk, like that's certainly not true. And so it's much more diverse than we originally anticipated. And so at least coming from basic research, perspective, we are trying to figure out these cellular factors that are involved, not just from the design perspective, right? But, in order to develop better vectors, right? One thing we can do is to fix the gene of interest, but the other is maybe perhaps we can find producer lines that can overcome these issues. And so I think I do share some of the same limitations that you guys are finding in manufacturing as well as transduction efficiency. So,
since we provide the sequencing technology, this is a question that I'll maybe just answer from that perspective, because we're really interested in helping all of the end users get accurate and robust results. But what we need is to hear back from people. What's your experience? What are your hurdles that you have to overcome, so that we can then take that internally and continue to develop and collaborate with our partners like FormBio and Solid Bio here with Phil, but hopefully also you. So please, what our limiting block is somewhat the information just communicating back to us so we can continue to develop.
Yeah, I guess I would just add a little bit to what Phil and Nadia said, which is, we've talked a lot about vectors and the heterogeneity in the vectors. What we really haven't mentioned so much about is that a lot of it is also going to come down to the combination of vector and capsid that you use also. And that combination will also change the heterogeneity quite dramatically, likely. So trying to develop a platform is challenging in that regard, because a lot of times you want to get to a capsid that may target a particular tissue, and then you want to stick in whatever vector, and you think that every time you stick something in for a different indication, it should all work the same. And I think everybody should be prepared that it doesn't. So that's really the bottom line, and that the legwork has to be done each time you develop a therapeutic, and that it's going to be difficult to just have a single platform for everything.
So I will give a perspective from the software development side. So when I think about it, I believe having workflows, pipeline that are validated and that we can use to reproduce results is important. So we have a lot of solutions out there, and most of the time you cannot compare them because the process is different, the nomenclature is different, the alignment may be different, or even if you have the same aligner, the settings are different. So how do you compare all those different things? And that's a main challenge, to make sure that the result that we are providing to basically the customer, the communities, are in alignment. So something that was interesting when you think about the other fields is Genome in the Bottle and the NIST. They put in place programs where you can order some materials and test your workflow. So you have a vector that have been characterized really well, so you know what to expect. So when you have a new tool, a new technology, you can just go and compare it and see, am I doing well, am I doing better? And that's something that is really missing, I believe, from the field. So how can we work maybe with CDMOs and our collaborators here to create publicly available data set that we can trust, and make it just easy to compare how well you are developing, how well you are doing. And I believe there is a lot of things that we'll be able to do there. So for FormBio as well, another aspect of it is the quality of the data, because as we develop all those models, understanding that the data that basically we are collecting has the right information is important. So rather than just thinking about the software side, going upstream a little bit on the manufacturing side of the things, the library prep, the sequencing, and create standards and best practices that basically we can use to reproduce those results and make sure that we have confidence in the data.
I can think about a couple of challenges that we're facing today. One, we talked about earlier, is just the construct design. But why that's important, it leads into the heterogeneity in the different serotypes and the different diseases that we go after, both non-rare and rare. The same serotype or a couple serotypes are going after both rare or non-rare. So we can't promise that the same serotype is going to provide that output. And those are the biggest challenges we face in how we work with the clients, because that leads into the second one, which is the cost of goods. because one of the major inputs into a process are plasmids. And the more batches, the larger batches that we need to treat this population, we're increasing the cost of those plasmids. Fortunate for us, there's a few companies out there that are actually tackling this, and although it hasn't been proven in larger scale, they're working through creating those packaging and producer cell lines to help address this. But we absolutely need to think it through, like whether they're upstream, downstream aspects from a manufacturing process that are going to help us reduce those cost of goods and provide more outputs. And that's going to help the industry as a whole as we try to tackle a lot of these diseases. Lots of diseases that we haven't even heard about yet.
Super. Okay, so we heard a lot of standardization issues as a main challenge. So I want to zoom in on that a little bit and think about how do we, okay, so I have two questions that I want you to think about. I'm going to call people out instead of pass the mic because I want to curate this a little bit. But I want to think about the places where we need standardization today. And I want you to think about how can we be good stewards of doing that work for technologies that are yet to come. So let me be a little more specific. I'm going to start with you, Nadia, because what I would like you to address is thinking about, in particular, your hardware. Adam brought it up as well, how we move from an industry that's built on Sequel IIe data to an industry that may be built on the Revio. And so I want to hear your perspective first on how we think about standardizing each piece of tech and how we move through that as we evolve. And then I want to hear from Phil and Adam about how we think about that from the experimental side and how we fit that into drug development, please.
Thank you. And I also want to add, we currently have on market the Sequel IIe and the Revio, but, we also continue to innovate on our instruments. So to be future-proof, we also have to think of future instruments and future sequencing chemistries, right? So in terms of standardization, I think, say something like a standardized, well-characterized reagent, like what Alpha has mentioned, would really help us to be able to just compare across the different iterations of our technology and then ensure that it performs the same way or in a comparable way, and if not, find some sort of normalization around it, right? So we're still not really there that we have these kind of materials on which we could optimize our technology. So we're somewhat reliant on our partners to share their constructs with us, so we can compare them across different platforms and then work together and collaborate to try to standardize and to try to make sure it works across different platforms. So that's very much a work that's ongoing and that we're very interested and invested in.
Yeah, thank you. So I think it's a major problem, right? At least from an academic standpoint, because many times you publish a manuscript that describes contamination or truncation events, and then when other people can't repeat those data, it becomes a major challenge for us, right? And so the fact of the matter is like technology will always march forward, and there's going to be improvements to be that are made, right? And it changes the way that we process our workflows. And just to be clear, right, the AAV field and gene therapy field is probably a very small market when you consider all of the things that PacBio or Nanopore or even Illumina sequencing does, right? And so, we can kick and scream and say what you've changed in your platform is no longer compatible with what we do, but I think we also have to be realistic about our demands as well, right? And so if we work closely with PacBio and these other sequencing technologies, and we're fair with what we see, right? Then we can also march forward, right? But also at the same time, I think we also have to consider that when we're getting sequencing data, right? That if the data that we generate isn't reproducible from things that are generated 10 years ago, there needs to be a little bit of flexibility, right? Because I often get those emails, right? We can't reproduce your data anymore. And it's like, sorry, right? So, right. So, yeah, I think it's a huge issue and it's important to address.
Just one final point to go off of what Phil said. You know, there is one variable that is the sequencing instrument itself and how the data is processed. But the other one is the preparation of the samples, of the DNA extraction, of generating the libraries. There are many steps that, you know, can be handled in different ways. So it's very hard to compare sometimes if it's not really apples to apples.
Yeah, I agree with all of that. And I think it is, I think Phil and Nadia are right. It's hard to, keep up with technology. Maybe, I guess the only thing I could think is one of the ways would be to start to normalize between instruments as we see technology coming down the pipe, is figure out a way to, show reproducibility and make sure we don't understand what the level of variability is from run to run, and we need to understand that so we can understand exactly how tight the data can be and how tight the data will be, so that we can compare across those instruments, maybe using the same vector, and once we have that, we could probably normalize different instruments out, different processing out. That's my thought.
Okay, that's really great. Thanks. So basically, what I'm hearing from everyone is we need a standard set of reagents and constructs and data sets that we can use for everyone. That needs to be open source and able to use. Like Genome in a Bottle is a great one, so that we can use that to test all the things. Okay.
Sounds like AAV titer.
A little bit, yeah, hurts.
But then just the same way defining a titer changes with technology, right? Like, when these sequencing platforms get better, the workflows will have to change, right? So even if we do come up with a way of standardizing them, is it going to be always, a moving target, right? So I don't really know how to address that myself, but,
Right, so we need a way to standardize today, and we need a way to define how we standardize in the future, so that at the very least, we can follow the same process for each time we need to make a significant change. Okay, along the lines of the standardization, the other thing that I'd like Nithya to comment on is thinking about the standardization for the production side. Not just the actual process of it, but are there things that we can think about from an economic perspective that make it more accessible to do things like replicates, right? There are certain scientific things that we would normally do to address like biological variability and things like that are really hard to do when you're under the drug development pressure to get something into patients and under the immense pressure to produce at, therapeutic dose. So if you would comment on your thoughts on standardizing that part.
So if I get this correctly, are we standardizing, like, coming into a CDMO, like how we would, try to replicate what they've done on the bench scale?
Well, or just how do you get process to process repeatability? Because you can't do an N of 10 and do statistics.
Yes, that makes sense. Yeah, if we could do N of 10, great, right? But, that does take time and resources. Before we even go into the larger scale where we're thinking about going to the 50, 200, 500, one of the things we've thought about, and, we've talked to a couple people about this, but when we're bringing in your construct into the CDMO, based on the cost, sometimes we can only do 2 N of 2. That's it. And one fails, then you're like, oh, maybe the other one works. But it's a 50-50 chance at that point. But the only reason we do an N of 2 is because we're like, our scaled down model might be only at a 2 liter. And that's the way then that we scale up to the 50. There are definitely ways for us to standardize that a little more and ensure that what's being brought into a CDMO is going into the smaller scale and automating it a little more. We spoke to one partner that actually has a micro batch offering. And with that micro batch offering, they can go do a 200 microliter automated, screening, basically. So maybe it still costs time and money, but now you can have your biological replicates, your triplicates, and ensure that what you have going into that larger process actually makes sense. I think that's what we need is great, it fits into our platform, but what can we do before we go from the in silico modeling and the construct design into something larger and just verifying that what we have actually makes sense in our platform?
Great, thank you. Sorry for all the walking back and forth. Okay, so the next thing that I want to talk about, I want to zoom out a little bit from that and think about the FDA and what it means to do drug substance, drug product characterization for the purposes of getting ready for an IND and doing the IND filing. So the things that I'm interested in here are your thoughts on the different levels of resolution that we need on these vector genomes, and where in the design process we need very high resolution versus places where it may be more effective or important to do lower cost, lower throughput, right? How should we be thinking about the places that the different sequencing technologies live in the development process? Because we've got short reads, right? We've got different kinds of technology for long reads. They all have pluses and minuses. And I want to hear your thoughts on where those different technologies might fit in the development of either the research side or the drug development side.
I'm probably the worst person to answer this question. I do know that, the only thing that the FDA requires is using NGS to sequence plasmid, right? And that's a sort of a requirement. But in terms of, that's not true. Oh, I don't know. Oh, yeah.
So
maybe you guys can, yeah.
So from your perspective, then, when you're doing your development, you're using principally PacBio long read sequencing,
right?
Why are you doing that as opposed to the other tech? And then let's do the FDA.
Most of it's from an academic standpoint, right? Just trying to understand what the heterogeneity is, right? And whether we can understand a little bit about the biology, right? That's purely why we're interested, right? So.
Yeah, similar to Phil, like we're probably also not the best to answer this since we don't directly interact with the FDA. Why we believe biotechnology over the other technology is a good thing to use is that we want you to have confidence in your result. We want you to go there and be able to say, this was the best that we could do with the technology that was around today. This is what we could have known, and we're providing you with the data, right? How and which technology you choose, that is largely in your regulatory team's purview, I think.
I'm going to caveat this for the camera that I am not in the minds of the FDA. So, but I think from the perspective of the FDA, I could see sequence being used as identification purposes, for drug identification purposes primarily. I've heard that they are hiring teams there specifically for NGS, for long read sequencing technologies, to understand what the characterization is. I don't think there are no requirements as of yet as to how that'll be approached. I think going back to the issues of variability and reproducibility, those are going to be key aspects before anything can really be regulated, right? We need to know that these things are reproducible, that variability, understand that variability so we know what to expect, at least, on particular instruments, using particular processes. And it may be, getting back to an earlier question, it may be that some of this direction actually comes from the FDA, right? Some of how we end up going will be guided by their guidance, because that usually is what happens, right? They dictate something and everybody kind of falls in line and says, okay, this is what we have to do.
Okay, let me reframe my question a little bit, because I think we over-indexed a little bit on the FDA, and what I was really trying to think about is, for design purposes, we need really high resolution, really high genome resolution, so that we can find all of the places where we need to make sequence changes, right? I guess when we're thinking about characterizing our drugs, what are the main questions that you're trying to ask? So Phil's asking about the heterogeneity, right, from the perspective of like where the truncations are, what the flip and flop are. What are the main things that we need to think about from a drug product perspective? So it's going to be, right, chimeras, whatever, like this is the kind of thing. And then is the long read sequencing the best technology for that?
Yeah. So yeah, I think to build that then really is the biological connection to that, right? So, if you have partials in there, is there a biological relevance to those partials, or can we just ignore them? I mean, are they important? Why are they important? Is there a potency assay? In the potency assay, does something come out that we shouldn't have there, basically, in that assay? Or when we're in preclinical studies, do we see it? Do we see an improvement if we go to higher fulls from lower partials, right? And then we could start to match that up with some of the biological outcomes. I think that's going to be critical in saying whether any of this, there's a utility to it, right? I don't know. Who else?
Yeah, I think from a regulation perspective, NGS and long read will become more and more important because it will give you more information compared to the other assays that the FBA has been asking so far. So as an example, in a lot of cases, we look at full versus partial versus empty. And what is missing is what your capsid is full of, right? So what we have been seeing with the NGS and basically the PacBio long read, that those snapback genomes, they may have a length similar to your full length AAV, and you cannot really filter those out. So you are considering those full, but in terms of therapeutic effect, they can have very negative effects, right? And there is no other technology that can give you those type of informations. And based on what you are looking for, there will be probably in the future a combination of short read sequencing, long read, that will need to apply to look at the variation, for example. So long read can be interesting for structural variations, but if you are looking for deletions or substitutions at a lower frequency, the coverage of Illumina can be a better way to get those. So do we have a set of questions that we want to answer? We need to define that, and then based on that, what are the tools, the right tools that we need to use in order to answer those will be the next step. But all those technologies, they are very good, and then the consideration of turnaround time, cost, and others will be important in order to answer them.
Something that I've been thinking as we talk about, identifying, what's in our pool, would this technology then help us understand if any of these partials, not, or the different types of, basically not the ones we want, basically, can we predict if they'll have off-target effects? Because, that's another aspect, too. I don't know if we've characterized that enough in our release assays that any of these that are not our target vector, are they going to have any other harm? Because our AAVs are generally in vivo therapies. So we wanted, can we predict the safety of the therapy before going into a phase 1 trial where we are trying to think about that safety aspect? That might be something to think through with this long-range sequencing or NGS-based work where we can learn more about the product.
Okay, that's a great point. Yeah, the release for the product, that's a really important piece. And I think also we talked about RC AAVs earlier and some of the other possible toxicity problems. So maybe there is a place for the different types of sequencing technology for answering particular questions. So that's great. I have one more question that I want to ask the panel, and then I want to open the floor up to a broader discussion with our experts. So this is an easy one. I want to hear what you're excited about technology-wise. What's on the horizon? What's in bioRxiv that isn't peer-reviewed yet that you're excited about in AAV biology? It could be gene editing. It could be anything. What are you excited about?
That's a hard question, actually. So, well, I'm so one of the things I think remains unanswered is what the structures of the AAV, what are the structures of these vector genomes once they're delivered into patients, right? And so this is active work that we're getting into. When we first developed this technology, there were two things that were lacking, right? One is what do the genomes look like during packaging, which I think is an important question to answer. But then the other one is what happens with these heterogeneous forms or even intact versions, once they're delivered into patients, right? Are they persisting as we predict, as free-floating episomes within the cell nucleus, or do they integrate, right? So I think we can use these sequencing platforms to gain a little bit of insight into these mechanisms.
Thanks. I think we're really interesting. We find it really interesting, and I'm excited about seeing all the different emerging applications that are across the whole development process in the cell and gene therapy and biopharma world in general. Going really from target identification, like maybe even looking at RNA isoforms, through your development and research processes, through the manufacturing, and seeing not only developments in AAV genome sequencing, but say also things like insertion site assessment, gene editing outcomes, single cell assessments. So there's so much that's going on, and so many places where sequencing and long read sequencing in particular really adds to the process.
From my perspective, I guess I think looking at how, I mentioned earlier how different capsid vector combinations I think are going to be important, and I think it's going to be important to understand what transduction in preclinical models looks like and how these differ, and understanding how some of that heterogeneity in the vectors may relate to that, and then what that looks like following transduction. So if you can get single cell analysis and start to get an idea within a tissue, because there's a lot of heterogeneity within, we talk about, say, the heart, and there's multiple cell types there, and there's multiple heterogeneity within that tissue. So what does transduction look like there? When you take a chunk of tissue, you're taking out multiple different cell types, and just because you see transduction or you see a biodistribution to that, are you still changing that when you go? Is it fibroblasts more versus cardiomyocytes, for instance?
I think from a software perspective, it's mainly the AI and the ML tools that we can now use to speed up a lot of the discovery that we can do. So probably 2 years ago, nobody was using ChatGPT. We used to go on Google, look for something, read an article. And now a lot of us, we are just asking, and those different models are giving us a summary answering those different questions. And I believe for the field, it's really good to start seeing companies working on payload optimization, capsid optimization. We may be talking about expression predictions, immunogenicity prediction, and so on. So there will be a lot of different models out there that at some point we can use all together to speed up our understanding of those different drug products, rather than doing all those different experiments in a lab in order to get there. So I believe there will be a lot of opportunity there. There will be more and more data set available to train those different models, and there will be a shift toward trusting those different tools. It's pretty new, but probably in 1 or 2 years we'll have more opportunities to speed up at least the first iteration when we think about a new vector that we are designing.
Thank you. I think we're in an exciting time where we're seeing a lot more therapies, getting approved, CGT therapies out there. We're seeing, fruits of everyone's labor previously coming true. So it's really exciting to see all these new emerging technologies that are coming in to, speed up the process. I think it's great that we are now looking at more upstream from the clinical trials to see how we can characterize and learn about the products, and more particularly even thinking about NGS. I am no NGS expert. I think I need to start learning more. But NGS is definitely a tool that's not only helping us characterize the product upstream of going into a patient, but also probably downstream as well. And I can see this in the application just across not just vector-based gene therapies, but also cell therapies and biologics, as a way to ensure what we're putting in there is safer. So we're not seeing those events or anything as we get into humans. I mean, we don't want to see any harm for our own fellow humankind, right? So it's really exciting to see all these new emerging technologies coming in for us to characterize and provide safety profiles before we go in and test.
Awesome. Thanks, everyone. So at this point, that was all of my questions. I want to open it up to the audience.